So let's wrap up 11.3 by talking about how we can screw things up chromosomally. So if we're going to look at chromosomes to try to figure out what's gone wrong, we have to have some way of analyzing what chromosomes are there. So we do this by doing a karyotype. Now a karyotype is going to be this image taken at metaphase that shows the chromosomes that are present, and typically too will show the homologs or the sister chromatids attached that are present as well. So looking at this, you can see that there's the two homologs for chromosome 1. We can tell it's chromosome 1 because they stain with the same pattern. That's what you're seeing here with the colors. You can see the centromeres in the same location, and they're the same length. You can also see that these have their sister chromatid attached because you can have that kind of X pattern because it's two copies that are currently stuck together. Now we have multiple different types of chromosomes, each with their own unique staining pattern, banding patterns, centromere location, etc. And so in humans you can see the 23 sets. Now that last set we've talked about before will be a little odd sometimes because it's a sex chromosomes. In this one it looks pretty normal because this is a female. If this was a male you'd see like a little stumpy guy that'd be here labeled Y. So it'd be obvious that they're not the same. It's not like these ones where they pair up nicely. You wouldn't get that if it's a male. Now, by looking at this, we can analyze and see if there's extra chromosomes, and we can sometimes too see if there's like extra pieces on a chromosome. Because sometimes when we screw up crossing over, we'll accidentally add like both pieces that busted off that we're supposed to swap. Sometimes they both attach to the same one. So a chromosome can be missing a chunk, it can have an extra chunk. The chunk can flip upside down before it attaches. Sometimes you'll get crossing over occurring between two different types of chromosomes, and so that obviously is not good. So this is some ways we can analyze if things have gone wrong chromosomally. Now another characteristic of chromosomes that we can look at is the ends of chromosomes will typically have this very condensed region that kind of sticks around. So we can normally see that the tips of the chromosome, the tips of the DNA pieces, seem to stay condensed consistently, and we call these the telomeres. So it's the end of the chromosome, condensed region, and this is significant because each time that the cell divides, each time the cell replicates its DNA, it typically loses a small little chunk off that telomere. So that telomere is kind of a gauge for how many cell division cycles a cell has gone through. And as it gets smaller and smaller, this can kind of trigger some of our cells to realize that something is going on where it's dividing too much. And so to try and prevent things like cancer, where we have these issues where you divide uncontrollably, we have built-in things that try to make cells that have this telomere shrink too much go through apoptosis to make sure they don't get up to mischief. We also have an enzyme called telomerase that can rebuild these end pieces, because otherwise you'd give birth to a child, and if you're already 30 at the time, you don't want your child to come out with telomeres that are a 30-year-old's telomeres, and then ultimately they keep aging. So we do have this ability to fix our chromosomes, if you will, where we rebuild these telomeres back to their normal length. So it's not the small version, it gets to be the large version, but this can also be fraught with issues because it's meant to shrink to kind of give us this record of how active that cell has been at dividing. So if you just keep throwing around telomerase like crazy, that can help encourage things or at least make us not notice things like cancer. So it's kind of this double-edged sword of, yes, you age in large part because your body realizes that your cells have gone through a lot of cell division, and so it starts to slow the cells down. And in some cases, they can actually get destroyed if they've reproduced enough times, and that makes your skin get thinner and age. But you also have, whereas time passes, we're more likely to build up the mutations that are necessary within a single cell to get cancer or something, so I don't really know which way you're supposed to fall on this. But some organisms, like lobsters, that don't really seem to age, do produce telomerase, so they seem to bypass this, and they just allow their body to think that it's kind of young forever. I don't know how often you see cancer and such in those species, but at least for us, the lobster route doesn't seem like it'd be that effective. You'd still likely die at a pretty normal age from a whole bunch of other things. Although I guess maybe in the future we could at least die looking younger at that age, which, that's cool. Now, non-disjunction is when we screw up the process of separating the homologs or separating the sister chromatids. Now, non-disjunction, if it happens with all the chromosomes, can lead to being a polyploid, where you've got a complete other set, so you're 3N or 4N. But probably more frequently, what happens is this occurs with a single homolog or a single sister chromatid, and so you accidentally grab both homologs, or you accidentally grab both sister chromatids. And so you're not up an entire set of chromosomes, you just have one extra chromosome, or you're missing one chromosome because you didn't get to grab any because the other guy did. And so this will lead to something where you're essentially 2n plus 1, or you could be 2n minus 1. Now this is a particularly big deal because if you have an extra or missing chromosome, this leads to a bunch of disorders. Now most of these disorders are bad, they're fatal, not cool. But there's a few disorders that you can survive, and some of them even have minimal impact, especially the ones that revolve around the sex chromosomes. So an example here is if you have 3 copies, so it's called trisomy, just because you have 3, 21 because it's of chromosome 21, that's Down syndrome. So ultimately you tend to be a bit shorter, tend to have certain facial features, some degree of mental retardation, sometimes you have heart issues. So there's a bunch of fairly negative things that you get if you get Down syndrome, but it's caused by an extra chromosome 21. There's also Turner syndrome as an example, is where you're sometimes written as XO. And the idea here is you only got one X, so you're missing the other sex chromosome. So in this case, you tend to be shorter than usual. Sometimes you're born with like a webbed neck. Very mild mental retardations. We're talking just a bit of a dip, but nothing terribly severe. And you oftentimes will have fertility issues, so you would not be able to have kids. So those are 2 examples. We're going to get to some more examples with the last slide though. And as I said, most of them are going to revolve around the sex chromosomes because that's the one that's easiest to mess with and still live. Most of the other ones, if you get trisomy 13 or 18, they just kill you either before birth or shortly after you're born. So wrapping up here, some of the things that you can get that you can live with. For females, XX is normal, we've talked about, but we already said with Turner syndrome you can survive and live a relatively normal life, albeit you won't be having kids, if you're missing a second sex chromosome, so just X. You can also have triple X, which might have minor things. There's been some things I've seen that they might have very, very slight mental retardation, but nothing that's going to stop you from living a normal, productive life. But otherwise, they're sometimes called triple X or super females. So these pretty much function like females. You probably wouldn't know you had it even if you did. Now, it's relatively rare, so don't just go around thinking, oh, I must have that. No, you probably don't. But it doesn't have any serious repercussions. And I believe when you pass it on, it doesn't even typically pass on if you have offspring. Now, for males, we've got our normal males, and then you've got Klinefelter syndrome, which is going to be XXY. This is what sometimes people confuse as like hermaphrodites. Oftentimes what you are is just a male that tends to have a bit more effeminate qualities. You might have some more female secondary sex characteristics, so it just means you might have a little bit different carrying of your body weight. You might have be a little bit more chesty, things like that. Oftentimes have fertility issues once again because you're kind of not producing as many male hormones, so you don't typically as high of like a sperm count, so it can make it more difficult to have children. But ultimately, you can live a relatively normal life. I mean, this is not like it's going to be completely disabling. And then there's XYY, sometimes called the super male, which doesn't appear to have huge complications other than I've read that a disproportionate amount of people that are XYY seem to end up in prison. So perhaps it's producing a bit more testosterone that makes you take a few more risks that aren't always completely thought out. So beyond just perhaps being a bit too aggressive for your own good, not thinking things through quite enough, and possibly spending some time in the penal system, you still can be a person that lives a normal lifespan, and I believe in most cases these individuals can reproduce, and I do not believe that they pass it on. So typically they'd have offspring that are normal. So the upshot of having karyotypes, a way to see the chromosomes, of knowing what disorders are caused by which chromosomal abnormality, we now can go through and do fetal testing. So depending on the exact timing, if it's very early in the pregnancy, you tend to use what's called CVS or chorionic villi sampling, where they take essentially a little piece of what is the placenta. You can also go through if it's later and get an amniocentesis or actual fetal blood sample. Either way, we get cells from the fetus, so we can go through and do these genetic tests to see if there's chromosomal abnormalities or other genetic disorders we can test for. Now, in some cases, if we know about this, that can allow parents to make wise decisions. That could be just preparing for things. In some cases, you could have where people terminate the pregnancy. You can also have cases where if the child has something wrong with it, they can do surgery even inside the womb in many cases. So this is kind of a really cool but potentially dangerous thing where they can now go through and try to fix some particular issues before the child's even born, which allows a child to survive that otherwise might not.